Hey everyone, and welcome to today's Explainer. Today, we're diving into Chapter 4's breakdown of genetics, putting a big spotlight on the principles of inheritance and variation. If you've ever looked around at your family, noticed certain traits being passed down, and wondered about the underlying math behind all of it, well, you are definitely in the right place. We're going to break down the actual mechanics of this biology using some really clear step-by-step concepts. To really ground this complex science, let's start with a fundamental biological mystery right from the text. Think about it. Why does an elephant always, without fail, give birth to a baby elephant and not some other animal? Or why does a mango seed only ever grow into a mango tree? It sounds super obvious, right? But for thousands of years, the actual biological mechanics guaranteeing that continuity were a complete mystery. Today, we're looking at exactly how life pulls that off. So here's our roadmap for today. First, Mendel's laws of inheritance. Second, exceptions to the rules. Third, the chromosomal theory. Fourth, linkage and recombination. And finally, number 5, mutations and genetic disorders. Let's get right into it. Okay, starting off with Section 1, Mendel's laws of inheritance. We're going to travel back to the mid-19th century, to the era where these foundational genetic rules were first established. But before we look at the experiments themselves, we need to define our absolute baseline terms. Inheritance is simply the process by which characters are passed down from parent to progeny. It's the whole basis of heredity. Variation, on the other hand, is the degree to which that progeny actually differs from its parents. And fun fact, humans were actually exploiting natural variations to selectively breed plants and animals way back in 8000 BC. But, they didn't actually know the exact mechanisms behind how it was all working. That is, until Gregor Mendel enters the chat. Between 1856 and 1863, Mendel conducted all these hybridization experiments on garden peas. Now, what made Mendel an absolute pioneer wasn't the peas themselves. It was his completely revolutionary methodology. For the first time ever, he applied statistical analysis and mathematical logic to biology. He wasn't just throwing ideas at the wall, he used massive sampling sizes to give his data real credibility. He investigated characters that manifested as two very clear opposing traits, like tall versus dwarf plants or yellow versus green seeds. By treating biology like a math problem, he started finding the general rules. Now, what's really fascinating here is how a test cross visually decodes a plant's hidden genetics. Say you have a violet flower. Violet is a dominant trait, but you don't know its exact hidden genetic makeup. Is it purely dominant, or is it secretly carrying a hidden recessive white trait? Well, by crossing this unknown dominant plant with a purely recessive white plant, the resulting offspring literally reveal the truth. If half the new flowers pop up white, boom, you instantly know the parent was carrying that recessive trait. It is just such an elegant mathematical proof. So, from all these crosses, Mendel laid down two foundational principles. First up, the law of dominance. Discrete units, which he called factors, occur in pairs. If the pair is dissimilar, one member dominates the other. End of story. Second, the law of segregation. These paired alleles do not blend together. When the organism forms gametes, the alleles cleanly segregate, meaning each gamete only gets one. No mixing. Which brings us to Section 2, exceptions to the rules. Because, as we all know, nature absolutely loves to throw in some rebellions. Biology is rarely perfectly clean. Mendel's law of dominance basically assumed one trait always wins. But look at this contrast. In incomplete dominance, we actually see an intermediate blend. If you cross a true breeding red snapdragon with a white one, you get a pink one. Neither completely dominates. Then, on the flip side, there's codominance. Here, the offspring resembles both parents at the exact same time. The ABO blood grouping in humans is a perfect example of this, where different sugar polymers on red blood cells can be expressed simultaneously. So dominance isn't some autonomous feature, it heavily depends on the actual gene product being produced. Now, when Mendel leveled up and studied the inheritance of two characters together, like seed color and seed shape simultaneously, he discovered the law of independent assortment. This basically means the segregation of one pair of traits is completely independent of another pair. And if you map this all out on a Punnett square for a dihybrid cross, it reliably spits out a super famous phenotypic ratio, 9 to 3 to 3 to 1. That's 9 round yellow, 3 wrinkled yellow, 3 round green, and 1 wrinkled green. The math just scales up beautifully. Moving right along to Section 3, the chromosomal theory. Okay, so Mendel gave us these flawless mathematical rules, but there was still a glaring physical mystery. Where exactly were these factors living inside the organism? See, Mendel published his work in 1865, but it sat around largely unappreciated and unproven physically for decades. Why? Simply put, our microscopes just weren't good enough yet. It wasn't until the turn of the 20th century that scientists could actually physically observe cell division. By 1902, Walter Sutton and Theodore Boveri finally worked out the physical movement of chromosomes during meiosis. The physical proof had finally caught up with Mendel's abstract math. Sutton and Boveri made this absolutely brilliant, paradigm-shifting deduction. The behavior of chromosomes was entirely parallel to the behavior of Mendel's genes. Think about it. Chromosomes occur in pairs, just like Mendel's factors. They separate during gamete formation, exactly like Mendel's factors. Sutton basically united chromosomal segregation with Mendelian principles to create the chromosomal theory of inheritance. Chromosomes are the literal physical vehicles carrying those abstract genetic factors. Let's see how this builds into Section 4, linkage and recombination. Because discovering the physical vehicle immediately created a brand new scientific complication. To verify this new theory, Thomas Hunt Morgan started experimenting with the fruit fly, Drosophila melanogaster. Why fruit flies? Well, they breed super fast, you can easily tell the males from the females, and you can see their hereditary variations under pretty low-power microscopes. Morgan was tracing dihybrid crosses for genes located on the X chromosome. But when he did the math, the results did not match Mendel's clean 9:3 to 3:1 ratio at all. He found significantly higher proportions of the original parental gene combinations than anyone expected. So the crucial point here lies in physical proximity. Mendel said traits sort independently, right? But Morgan realized that because these genes literally live on a physical chromosome, if two genes are physically close together on that same chromosome, they tend to physically stick together during reproduction. He coined the term linkage for this physical association. Linkage basically breaks the independent assortment rule because the genes are physically tethered together, which leads to a really low rate of recombination and a high proportion of offspring that look exactly like the parents. Finally, we've reached Section 5, mutations and genetic disorders. We understand the code, we know how it moves, but what happens when the very structure of this code changes or breaks? Well, this brings us to mutation. Fundamentally, a mutation is just a change in the genetic material, and sometimes it is astonishingly small. Like, a point mutation is a change in just one single base pair in the DNA strand. But that microscopically tiny change can have massive physiological consequences. Sickle cell anemia, for instance, is caused by the alteration of just one single base in the gene coding for the beta chain of hemoglobin. Just one altered base pair completely alters the entire phenotype of the red blood cell. It's wild to think about. And mutations aren't just limited to single tiny points. Sometimes there's a failure in the segregation of the chromosomes themselves, leading to a gain or loss of an entire chromosome. This makes the chromosomal theory incredibly, starkly real. Down syndrome occurs when there's a whole extra copy of chromosome 21. That's known as trisomy 21. Turner syndrome occurs in females when an entire X chromosome is just missing. Conversely, Klinefelter syndrome happens when there's an extra X chromosome. These structural changes fundamentally alter human development. Which brings us full circle to a really profound conclusion from the text. Awareness in the areas of molecular genetics have enriched our understanding of the molecular basis of evolution. You see, those variations and mutations we just talked about, they aren't just anomalies or mistakes. Over millions of years, they're the literal drivers of evolution. The microscopic variations in that chromosomal code are exactly what eventually created the elephant we asked about at the very beginning. It's one continuous chain of inheritance and variation, which leaves us with one final fascinating thought. If errors are the true engine of evolution, what invisible, microscopic mistakes occurring in our code right now will define the next giant leap in human development? Something to think about. Thank you so much for joining me for this explainer.